So the question was the difference between a chromosome and a chromatid. Come on in. And the short answer is they're the same thing. And we're going to go through this in detail in class. So I'm going to give you a short answer. If you want to ask more follow-up questions now, that's totally fine. But I don't expect you to know this material yet. So there's a chromosome. There's a sister chromatid. Same thing. So how are they different? I mean, why do we have two names for the same thing? That's the real question to ask.
My understanding could totally be wrong. The chromosome is a larger mass or a collection of the chromatids.
So when we look at something that's a chromosome in, oh man, I wish I could spell, metaphase of the cell cycle, right? That's part of meiosis and mitosis. When the cell is about to divide, the chromosomes replicate. That's synthesis. Boy, maybe time for a new synthesis. Then you get mitosis, which includes metaphase. See something look like that we call a chromosome, but we just discussed that's really two chromosomes. It's two copies. Let's say this is humans we're talking about. So that's the version of chromosome 1 you got from your mom, one maternal, and a copy of chromosome 1 from your mom. And then you'd also have in the same nucleus the same chromosome, chromosome 1, but these are versions from your dad. So one paternal and one paternal. Okay. And that's 4 double helices. 1, 2, 3, 4 double helices. And these are called sister chromatids. So those are 2, I'm going to abbreviate SC, and there's another 2 sister chromatids.
Okay, so when it's just by itself, the double-stranded DNA is, you can call like a chromosome.
Right.
And after it goes into metaphase, it'll duplicate itself.
Yeah, synthesis happens, that's the replication. And it's, so you're getting there, keep going.
And then it's going to go, so they'll replicate it to have the sister chromatid, and then in metaphase they'll condense into the sister chromatids.
What we see as chromosomes, yeah. This is the only time in the entire cell cycle, metaphase, is the only time we actually see DNA that looks like what we consider a chromosome. If you have a mental picture of what is a chromosome, those shaped things, that's the only time that they look like that in the cell's entire life.
Okay, but looking at the chromosome as we see it, it would be improper to call it a chromosome. It would be more proper to call it a sister chromatid. They're
only called sister chromatids between synthesis and cell division. And I don't care if you know that. I want you to know that if you want to know that, but I'm not going to test on that in class. So between synthesis and cell division, that's when there are sister chromatids. But the really important point is that there's no actual physical difference between a chromosome and a sister chromatid. They both refer to a double helix.
I was just confused because they would use, the terms back and forth.
Yeah.
And I don't know really what
right.
Exactly.
So the definition, right, is that a sister chromatid is a chromosome between synthesis and cell division. During most of the life of a cell, the 99% of the time it's not doing mitosis, there are 2 chromosomes. 2 chromosome 1s, 2 chromosome 2s. But after replication, after synthesis, when there's 4 copies of chromosome 1, those are 4 sister chromatids. And as soon as the cells divide and we go back to 2 copies of the chromosome per cell, because after cell division, of course, the point of mitosis and cell division is that at the end you get a copy of 1 from your dad and a copy of chromosome 1 from your mom in each of those daughter cells, so that they're genetically identical to each other. So here they're chromosomes. It's only when, and this is related to something that I don't think I've asked you to watch a video on yet, but will soon. is ploidy. How many copies of chromosomes do we have? Humans are diploid. We have 2 copies of our chromosomes. Well, that's true here. Right? Every cell has 2 copies of chromosome 1, and then also 2 copies of chromosome 2, and so forth. What are we at metaphase? How many copies of each chromosome? How many double helices of chromosome 1? 1, 2, 3, 4. We're tetraploid between synthesis and cell division.
Is that going to be closer to the section where you start to look at genetics, whereas polyploidy?
Right. Yeah. So if you take anything away from this, it's not necessary. It's 2 things. One is that chromosome and sister chromatid refer to the exact same type of molecule, a double helix. The only difference is one term is used for part of the cell cycle, the other term is used for the other part of the cell cycle. The second thing to keep in mind is that when we talk about ploidy, copy number, how many copies of a chromosome, we're always talking about interphase pre-synthesis cells. That is, how many copies of each chromosome, say chromosome 1, does a cell have before it replicates? Because we have to have some sort of definition. Because we're diploid until we're tetraploid. Organisms that are octoploid have 8 copies of every chromosome, are octoploid until synthesis happens, and they're hexadecaploid. You've got 16 copies of every chromosome. Right? So geneticists just had to have a fixed reference in time for saying this is when we're going to define ploidy, how many copies of every chromosome an organism has. And we decided that it was going to be interphase of the cell cycle pre-synthesis. And that's when ploidy is defined. How many nucleotides long is this sequence? I got 26. Let's just say it's 26, for the sake of argument.
Okay.
So 26 nucleotides, you've got how many A's? 4. And the rest are T's, G's, and C's, and so forth. So, do we have to write in the second strand to figure this out, or can we just do this calculation from the single strand?
Single strand?
You could do it from. That's what I. So
So
since with the addition of the T's and the G's, it increases the overall amount to.
So let's take the first 8 nucleotides. AACC, AAGG.
Gotcha.
So that's 50% A?
Mm-hmm.
Okay. What happens to the percent A when we write in the other strand?
Still the same.
Well, we've got more nucleotides, though. So
Got
instead of
16
having
times.
8 nucleotides, now we've got 16.
So it drops to half.
So it's now we've got, so one strand gives you 50% A. If you look at 2 strands, now you've got 25% A. So we need to know the sequence of both strands. So up here, you've got 4 A's, and you've got how many, so that just tells us we need to count how many T's. 1, 2, 3, 4 T's. So out of 26 nucleotides, what you've submitted here, 8 of 26 are A plus T. That means if we wrote in the second strand, if we were looking at the full molecule, that would be looking at, instead of 26 nucleotides, now we'd have 52, and because of base pair complementation, when you have 8 A's and T's, that means that when you look at this other strand, there will also be 8
Double.
A's or T's, so that's going to be 16 out of 52. That's A plus T. So what is that fraction?
16, 52, 30%.
So that's 30% A plus T, which means that your percent A is half that.
15.
Yep.
So I guess what I'm trying to ask is really this: if I were to give a just a random code that you asked for, and
Yep.
the A's don't necessarily need to match up to, like, say, the T's within the same strand. Obviously, the
Correct.
opposite side is going to have T's to match it anyways.
Right.
But,
Yeah, so you don't have to write the second strand, and you don't have to have an equal number of A's and T's,
okay.
as you've done here. Which, yeah. You just have to have, so for example, this still would work if this, which was one of the examples that we did in class, this strand doesn't have to have any A's in it.
Right. It could have.
It
just
could
the
be all
T's.
T's.
Yeah.
Because we know that on the other strand is where the A's are, and those are the A's that are going to comprise the 15%, or
Gotcha.
whatever it was that we're looking for.
Yeah. Okay.
So you could have all T's and C's, and you'd know that the other strand is where the A's and the G's that I'm
Right.
asking about are going to be located.
Okay. That
Yeah.
that was my
Yep.
the ambiguity that I was trying to.
Yeah. Let's see, so we're talking about RNA base pairing. Anything that's in the stem is going to be complementary. So if you've got some sequence here, A, T, C, G, if it's the stem, by definition, there's going to be the complementary bases on the other side. And the only reason that these residues up here, these nucleotides, form a loop is because they don't have, across from them are not nucleotides they would normally base pair with. So for example, if there's a G there might be a G there. And so they're not base pairing, they're forming a loop because there's no possibility of hydrogen bonding. And they don't have to be mirror images of each other, so it could be A and G, which can't base pair with each other. And when I drew that example, I just knew that the easiest thing for me to do in class was to put the same nucleotide all around the loop, because nucleotides don't base pair with themselves. So I just put U all the way around.
Okay, and that's also where the hydrogen bonding occurs?
And so uracil can base pair. In fact, these T's, since this is RNA, should be U's. So there'd be 2 hydrogen bonds there, and 2 there, and 3 there, and 3 there. And then nothing in the loop.
Just because it's a loop, it's in the
And it
hydrogen
just-
structure?
Just because that C can't make any hydrogen bond with the nucleotide it's directly across from. If that was a G, if this nucleotide there was a G, which I can't write, then that stem would be 1 nucleotide longer because the C would base pair with the G. But G then doesn't base pair with G, right? So there's no possibility of base pairing there. It's the same nucleotide, and A doesn't base pair with G. So that's the loop is formed by nucleotides that don't hydrogen bond with each other. Yeah.
Oh yeah, I have a quick question about the book. I was reading the book and I didn't